Hi everybody, we're going to take a look and fill in your notes on chromosome mutations and gene level mutations. So chromosome mutations are going to be, and let me get to the next slide, the goal for this is to determine how the mutations alter the chromosomes and how they alter the DNA sequence and how they can impact the phenotype or the physical traits of the organism and how maybe they won't. In general, and you can add this to your notes as well, mutations are heritable changes of genetic information. So this means that it's a change, it can be in the nucleotide sequence, if it's gene level, or if it's chromosome level, then it impacts the entire structure of the chromosome, which typically impacts a ton of genes. So chromosome mutations, again, if you change the entire chromosome, whether it's through non-disjunction because you have an extra one or an error in crossing over, then you can have extra information or missing information. Chromosomes contain a ton of genes, so if there's any errors on here, it's really detrimental to the organism because then it's missing hundreds of genes potentially as opposed to maybe an error in just one. Make sure that you add the causes of these in your notes as well so that you have those written down. The other mutation that we'll look at is a gene level mutation. So this is a mutation in the actual DNA nucleotide sequence of one single gene. It can also be really bad. I mean, you don't want any sort of mutations in your genes. It happens more commonly. Most of the time it happens with DNA replication, but this can certainly have a really significant impact on the organism. It just doesn't impact as many genes. It's just one gene at a time. So chromosome mutations, again, you're changing the structure or the number of the chromosomes, and then it might change the location of the genes or the number of copies of the genes. So maybe you have too many or too few. So there's 4 types that we're looking at. There's deletion, and so the picture in your notes shows this as well, but go ahead and put a little definition. So deletion is just like it sounds. There's a loss of all or part of the chromosome. With duplication, now you have an extra copy of all or part of the chromosome. This would happen probably because of an error in crossing over where it didn't happen at the right spot, and so then it has extra information. There's inversion, which means that part of the chromosome flips, so it's not in the right direction. So if it was supposed to be ABC, now it's ACB. And so the reason why that matters is because your body is set to read genes in a certain way, and so with the sequence flipped, it might not be able to read that gene. Translocation then is going to be part of a chromosome from a non-homologous pair is going to attach to another chromosome. So a non-homologous chromosome piece attaches to another non-homologous chromosome. Okay, we'll see this, or you could see this in karyotypes where the chromosome is extra long compared to the other chromosome, but it's the key to this is translocation is in exchange between non-homologous chromosomes. So that would be a problem with crossing over, which happens in meiosis, as you know, during prophase 1. All right, so non-disjunction, as a reminder, we're looking at this in meiosis as well. Non-disjunction is when homologous chromosomes fail to separate during anaphase 1. It could also happen during anaphase 2. It's going to cause gametes that then have the wrong number of chromosomes, and so then you can end up with something like trisomy, where you have 3 copies of the chromosome, or you can have monosomy, where there's only 1 copy. This is just a picture showing how chromosome mutations work and how non-disjunction would work during anaphase 1 and anaphase 2. And this is showing the same. So we're going to talk gene level mutations. So this is on your next page. Gene level mutations are going to be mutations in the nucleotide sequence. There's 2 main types that we're going to look at, and they're going to be point mutations and frame shift mutations. Point mutations are going to include things like substitution, and then we'll look at a few types of substitution. And then frame shift mutations will include insertion and deletion of nucleotides. Gene mutation errors or gene level errors are going to occur more commonly, and they typically occur during DNA replication. You're going to pass these along to the offspring potentially if this occurs during general development of the organism. All right, so gene level mutations, point mutations. So this is generally a substitution. It's going to be a point mutation that impacts one single nucleotide in a sequence. So one of those A, C's, T's, and G's. It's going to change the genetic sequence, which increases the genetic variability, which sometimes is a good thing. And it may or may not impact the phenotype or impact the protein. There are 3 types of substitution. So there's silent substitution, which is just as it sounds. There's a change in the sequence, but it doesn't impact the protein at all, or the amino acid produced. This is because of that codon chart where we have lots of codons that can code for the same amino acid. So small changes might not be even detected in the amino acid. We have nonsense substitutions, and so this is a point mutation that results in a premature stop codon. So there's some change to the nucleotide sequence that makes a stop codon, like UAG, I think is one of the stop codons. So maybe it did that, so now instead of the protein being 200 amino acids long, now it's only 20 amino acids long. The last type is called a missense substitution. It's going to be a change in the DNA sequence, and it's going to change the codon to a different amino acid. Sometimes they have an effect, but sometimes it doesn't. It just depends. But the key here is that the missense changes the amino acid. So silent doesn't change the amino acid, nonsense premature stop, and then missense, it's going to change the amino acid, but it may or may not have an effect. The other types of gene level mutations, here are some examples of substitution. And we'll do some of these in class as well. But if it was a silent substitution, like the bad cat saw instead of a lowercase a and uppercase a, like that's not going to impact it. But if it was something that changed the meaning, like instead of the bad cat saw, it's now the bad bat saw, it changes the meaning of the gene and of the protein that it makes. The other type of gene level mutations are called frame shift mutations. This is caused by either inserting or removing a nucleotide, and it changes the reading frame of the protein. So if you remember, whenever we did transcription and then translation was read in sets of 3 called codons. So if you eliminate one, then everything shifts over, and now everything's not being read with like its group. So this really can impact a gene and certainly has a bigger impact typically than just a point mutation. So frame shift mutations are really more negative for the organism than the gene level point mutations typically. So if a nucleotide is inserted and deleted and it's not in a multiple of 3, then the entire reading frame shifts. So I'll show you what I mean. Insert is you put it in, delete is you take it out. But so again, if the original sentence was the bad cat saw the big dog, blah, blah, blah. If you insert a CA, now it makes no sense. So after you have the error, now it makes no sense. So the protein that's being made by the organism makes no sense and isn't going to do the function that it's supposed to do. So now it says the cab and then just jumbled everything. So nothing's going to happen. Now, if you delete some letters, so now if you delete the BA, now you can only read the, and everything after the deletion is a jumbled mess. So frame shift mutations make everything a mess, and the protein now won't function like it's supposed to. We'll do practice with this, but again, the main types of mutations, we have chromosome mutations that are caused normally by non-disjunction and crossing over. We have gene level mutations, which can either be a point mutation, which is just a change in what the nucleotide is, or it can be a frame shift mutation, which actually changes the entire reading frame for the gene and then for the protein, so then the protein doesn't function. So if you have any questions, write them down and we will talk about them in class.